thanks everyone for being here and online. Today we're really excited to have David Freedman, Professor of Neurobiology at the University of Chicago. We're really excited to hear what you have to say.
Great, thank you so much. Let me turn down my sound here. That'll echo. All right, great. Well, thanks for coming, and I'm excited to come downtown on this beautiful day, tell you about the work that we're doing. So the main question that drives much of the work in my lab is to understand how the representations of visual stimuli in the sensory areas of the visual cortex, of the primate brain, how is that read out in order to allow us to interpret what the things mean out there in the world around us. This is a question I've been interested in for decades, and it's become an increasingly exciting time to ask this question, given the new methods that we now have for recording ever larger populations of neurons across larger networks of the brains, not just focusing on one area at a time, but now recording from pairs or triplets or even 4 or 5 brain regions at the same time, to basically ask how is activity coordinated across multiple brain areas to guide visually based decision making. So just some examples from our everyday life. So if you found yourself in the market shopping for fruit, you need to make a decision. Which cluster of bananas is the right one for you? And there's actually not a correct answer, right? If you were shopping for your lunch today, you probably choose the ones on the right because they're ripe and ready to go. But if you're just shopping once this week and you want to have bananas for 4 or 5 days from now, you'll choose the ones on the left. So making an arbitrary decision like this based on the context of the behavioral situation that you're in is kind of fundamental to making effective decisions. Another example is the performance of this guy right here, a Major League Baseball umpire. So what he has to do is hundreds of times per game make a judgment about whether the ball that's thrown by the pitcher is within or outside of the strike zone, right? So the strike zone, it's not a concept that this individual was born knowing about. It's something that he learned, right, through months or even years of practice. This is actually data from Major League Baseball umpires showing their ability to categorize balls and strikes. And you can see that this is really a beautiful example of categorical, visually based categorical decision making, where the red points are the locations of the ball when it crossed home plate that were called a strike by the umpire. The blue points were the positions that were called a ball. And this is arbitrary, right? Major League Baseball sometimes actually changes the rules on us. Yeah. Correct. Yeah, those are errors, and there's actually a couple of of blue points that fall in the middle too. So those may have been unpopular calls, but by and large, these are pretty good calls. Mm-hmm. Many. Yeah. I wouldn't be surprised. This has not been a focus of my research, but there may. Yeah, somebody's at least made this plot in what looks like maybe MATLAB. Right? But okay, so I'm interested really in what is changing in neural circuits in the brains of not Major League Baseball umpires exclusively, but in our experiments, monkeys that we train to perform visually based decisions that are kind of akin to these sorts of decisions, where a continuum of sensory stimuli need to be classified into discrete categories that are defined by arbitrary rules that individuals learn. So since I know this is a diverse group coming from many backgrounds and only some coming from a neuroscience background, I thought I'd just give you just a little primer on the circuit and the brain systems that we're interested in, and also tell you a little bit about the methods we're using to collect our data. So imagine an everyday situation where someone hands you a coffee cup like this one. Well, you need to decide what it is you're going to do about it, right? And that depends on the behavioral context. So some amount of light that is coming in through the windows, through these lights, photons streaming in, a small fraction of them will bounce off the object at just the right angle that they'll enter your pupil and then stimulate light-sensitive cells in your retina. So the photoreceptors, this is actually micrograph image of the three main layers of neurons in the retina, which convert incoming light into ultimately into action potentials that wind up streaming toward the brain, turning into your experience of the world around you. So some stimulation with light on the photoreceptors generates action potentials that stream down the optic nerve toward first the thalamus, then the visual cortex, and then to this whole hierarchy of areas that generates your visual perception and also ultimately your ability to make decisions and perform actions. Okay, so in the visual cortex, so what I'm showing you here, this is a lateral view of, or it's a drawing of the rhesus macaque monkey brain. So in my lab, we work with rhesus monkeys that we train to perform decision-making tasks. What I've highlighted here, the network of areas within the visual cortex. So there's a network of a dozen or so visual cortical areas that kind of break down that incoming image into representations of the basic sensory features of images. Features like edges, brightness and contrast, color, texture, position, the direction of motion. You can think of other features, and most of these features are then represented in one way or another among populations of neurons in this network of areas in the visual system. So we can call this a feature representation stage, kind of a faithful representation of the features of the stimulus, but kind of independent of what the animal's doing, right? Even if the animal's anesthetized, actually, you can perform experiments and record from these areas while the animal's eyes are open and looking at a screen, and you'll find quite similar activity. There are some interesting differences, but most by and large representations are the same during passive viewing as during active vision and things in these areas, at least in the monkey. It varies in some smaller animals more broadly. Okay, so then after this representation of the basic sensory features of the image, there's a network of regions which receives inputs from the visual cortex and is involved not in representing the features of the sensory stimulus, but instead representing what that image actually means to the animal, right? Has this image been seen before? What's the identity of the image? What category might the image belong to? And this depends crucially on past experience, right? You weren't born knowing about coffee cups, right? You learned about coffee cups through experience, which has left some lasting impact on neural circuits, encoding that remembered information for the long term. And then, okay, once this image has been recognized as this particular object, one needs to decide what they're going to do about it. And this depends on the situation. Depends on past experiences, past events that have just happened. Did you just order a coffee and it's been placed down in front of you by the server, or was this placed in front of you, but the person next to you ordered it, right? You're going to elicit different action plans in those two situations. This depends on your level of motivation, all kinds of things like this. So this is the stage of response selection, which I'm highlighting the frontal cortex and the parietal cortex, interactions between these areas, which we think are involved in deciding which response to make in a situation like this. And then finally, there's the motor response itself, right? So this, I'm highlighting the motor cortex, again, parietal areas that would control that, that reaching movement to reach for that object. And generate the right pattern of muscle activations, make sure the hand is in the right orientation to actually grab the object correctly, and so on. Okay, I just spent several minutes going through this whole cascade, but actually in real life this happens very quickly, right? In a third of a second, you'll make a decision about whether or not to reach for this coffee cup in a situation like this. And so, yeah, Becca. Mm-hmm. Yes. So, well, the networks that are very important for driving your attention around, or even your eye movements around an image, would be in this response selection stage, and it's actually within regions that are highlighted here. So areas that are directing attention are really thought to be in that frontal lobe attentional network in concert with areas in the parietal cortex and also the oculomotor system that are actually driving your eye movements around. But those areas project back to the visual system, right? So if you're looking for Waldo, then and there's a lot of work on this from many laboratories. The red horizontally striped shirt, isn't that what Waldo has, right? So neurons in the visual cortex that represent horizontally striped things and red things, they actually are subject to an increase in their response gain. So you will find that those neurons, if they prefer the features that a subject's looking for, will actually become more responsive. And those modulations are known to be generated in regions in the prefrontal cortex projecting back to those feature selective neurons. So the feature representing areas that are representing horizontally striped things and red things, those are here. The control systems that are projecting back and modulating the response gain is up in this more attentional network feeding back. But this also raises this problem. It's like, well, does that mean you have to have another feature map up here for red things and striped things? Or else how does that region know which populations to select back in the feature representing areas? That is a bit of an unsolved problem, and how that selection happens really interesting, and is very likely happening through coordinated interactions across these populations, right? You can't just have a duplicated representation in every stage. It's inefficient. It's inefficient even in computational systems, but it's especially problematic in the brain because our heads need to be limited in size, right? So that we can be born, right? Yeah. Okay, so how do we address this question, right? So at what level do we want to go in and record from these neural circuits? So in my laboratory, we record from single neurons. There are, in the human brain, something like 80 billion neurons in the human brain. Of course, in the macaque monkey, it might be something like 5 billion or 10 billion, something like that. It'd be an order of magnitude less, I'm sure. We record from individual neurons using electrodes that we surgically implant into the brain areas that we're interested in. These electrode arrays now have, as many as hundreds or even thousands of recording sites on a single electrode probe, allowing us to record populations of maybe 100 neurons, 100 to 1,000 neurons at a time while animals are awake and performing these tasks. So and the signals we're recording from are action potentials. So these are like all or none electrical spikes that are generated by neurons, which travel down the axons of these neurons, triggering release of neurotransmitters at synapses at their targets. So it sounds like a problem, right? We're recording from 100, maybe 1,000 if we're lucky. If you're Lucas Pinto, you could probably record from more than 1,000, but that's in a mouse. So it's they have some really awesome techniques to allow them to do that. So it sounds like a problem, though. What can we say about how this whole system works from a subsample of hundreds of neurons? The answer is actually we could say a lot more than you might guess. That's why this is actually a sort of fruitful enterprise, is that there's so much redundancy in neural populations that we can actually decode what decision an animal is going to make with actually better accuracy than the animal has. We can actually predict what the animal is going to do in many cases and what they're going to report about a stimulus more accurately actually than the animal's own behavior. So there's a lot of redundancy in neural codes, which is quite fascinating and something we could talk about more maybe offline. Okay, so the basic behavioral setup, this is an old drawing of, an older version of the way these experiments work, but basically we bring monkeys up to the laboratory in custom-made monkey chairs. They are positioned in front of a display where we can present images to them. There's this spot of light that you see at the center of the screen. This is, we typically require animals to maintain their gaze on a single spot of light at the center of the screen to control their eye movements so we know where they're looking. That means that when we present a stimulus on the screen, we know which neurons in the visual system we're stimulating. And then we can present images up on the screen, and through daily training sessions, thousands of training sessions over the time that we work with an animal, we can train them to perform all kinds of sophisticated tasks. We can train them to perform memory-based decision-making tasks. We can train them to perform games like we played when we were kids, where you have a bunch of cards all face down and you have to find pairs that are matching with one another, right? Monkeys can learn tasks like that. And they can, and their visual systems are set up almost identically to ours, so that anything that we learn about visual encoding in the monkey brain translates almost directly to the human visual system. We can then use an array of different kinds of neural recording approaches to go in, as I mentioned, recording from hundreds to now approaching 1,000 neurons at a time. Some of these approaches actually are used in human clinical studies and clinical research studies as well, using FDA-approved electrode recording devices that are implanted in the human brain, which have been interesting for like brain-computer and brain-machine interfaces, for example. Okay, so let me tell you about this behavioral task that we have been working with for a long time in my laboratory. When I first started experiments using this paradigm, back when I was a postdoc, I did not expect to still be doing so 20 years later. But it's taken us into circuits of the brain that we weren't expecting, and has actually revealed something about which networks are recruited to perform kind of flexible abstract decisions. Okay, so what I'm showing you here is a depiction of directions of motion. So this visual stimuli we show our monkeys in many of our experiments look like the one I'm about to show you. High contrast motion patterns, right? So this was upward motion. That corresponds to this direction right here, upward motion. Well, what we can train monkeys to do over the course of weeks of training is to learn that directions of motion belong in one of two groups. And that these two groups are categories that we invented. Category 1 and Category 2, they're defined by this category boundary, this green dashed line here. And what this does is it creates these arbitrary groupings, sort of like balls and strikes, in which directions that are in different categories can be very similar visually. These are only 30 degrees apart, yet the animal needs to report to us that those are different. And directions that are almost opposite, right? They can belong to the same category. So through training, what the animals learn to do is to abandon their initial approach of just saying that, well, things that are similar go together, and learn that actually, for the purposes of the task that they perform, they need to divide this range of stimuli into arbitrary groups. Now, monkeys don't talk. And so we need to get them to report their decisions to us in some way. And we actually do this in a few different ways, depending on the particular experiment. This is the task we've used the most often. And what we do is the monkeys they have their hand on a lever that's out in front of them. And the monkeys will release a lever to report to us whether one of these motion patterns is a categorical match to a previously presented pattern. So a trial would unfold with a sample stimulus being presented, can be any one of these directions. Then there's a delay where the animal just needs to remember what it is he saw earlier in the trial. And then after the delay, there is a second stimulus. Again, can be any one of the 12 directions. So half the time it'll be in the same category, like this case. Half the time it'll be in the opposite category. Yeah.
How often do you vary the category boundary?
Very occasionally. So it takes the animals weeks to learn the category rule. And so in the experiments I'll tell you about today, these are all like very well learned categories. We've done experiments where after we've kind of understood something about what's going on in the neural populations we're interested in, we've retrained the monkeys on a, say, perpendicular boundary to ask, well, it was kind of a control experiment, but also we wanted to ask, well, how flexible are these representations? New recording approaches are coming online. Here, this is one of our problems. We're not just recording from the same neurons every day for weeks and weeks and weeks. Every day we will implant electrodes and then remove them at the end of the day through a little chamber that we implant. And we have these motorized microdrives, lower the electrode down and remove it. That means we're looking at different neurons every day. What we'd really like to do is track neurons across weeks or even months as we're teaching the animals something. This is something actually that you can do with imaging approaches in smaller animals, particularly. Those techniques are going to make their way into the monkey soon, hopefully. But yeah, that'll open up the door to studying learning in a really cool way. So anyway, what the monkey needs to do is release a lever if the second stimulus is the same category, keep holding the lever if it's a different category, and then on these non-matched trials, a match image will show up out here as a third image, and the animal then has to release the lever. Yeah. Sure. Yeah. It basically initially completely arbitrary. They were I guess the task design though was chosen in order to satisfy some demands that we had. So we wanted to separate out the animal's decision about the category from the motor response that they're going to make, because we knew. The whole plan for this experiment from the beginning was to record from areas that were sensory and motor. Right? We knew we were going to record from areas that can encode what the stimulus looks like, also can encode something about the animal's motor response and decisions. So we want to know if we see signals that are correlated with category, we want to be able to say that's because of a contribution to the visual decision process, not the movement planning process. So that's why we separate out the decision about category from a decision about match non-match. That's because this task requires the animal to first categorize that sample, then keep that information in short-term memory. Then the animal has to categorize the test stimulus, compare it with the previous one, and then decide whether to release the lever. We've also we have several variants of this. We have tasks that are reaction time tasks where the animals immediately report their decisions by making an eye movement to one of two targets. And those were like very fast reaction time based tasks. So we're kind of branching out into other paradigms. Okay, so what brain network transforms visual features into abstract categories? Where we're interested here in visual motion, right? We're using visual motion as stimuli. So what are the areas that are involved in visual motion representations? Well, the first place to think about is visual area MT. This is an area that is critically important for computing the direction of motion of any visual stimulus. Some single moving dot, gratings, patterns, dots, textures. The direction of motion of that stimulus is really thought to be computed in area MT. So, we can argue that we have a better understanding of the representation of visual motion than any other complex visual feature. So it's a great place to start in terms of asking, how do you convert a directional or a simple sensory representation into a more flexible learned decision relevant representation? The other area we were initially very interested in was the lateral interparietal area, an area in the posterior parietal cortex that anatomically gets a strong input from the motion processing system, but is also, there's a long history now of studying LIP in the context of simple decisions. So we speculated that LIP is likely important for reading out motion information in order to generate these decision signals. Well, then over the 20 years or so, we've recorded from an increasingly broad network: dorsolateral prefrontal cortex, the medial superior temporal area, which is another region involved in motion processing, parietal area 5, which is an area that's involved in guiding reaching movements and hand movements. We thought that could be important given the animals are reporting their decisions by releasing a lever. But interestingly, in just the last few years, we have been focusing more and more on the brain, on the oculomotor system of the brain, actually the region of the brain that is generating these kinds of top-down projections you asked about that are modulating sensory representations. This network is also crucially involved in generating your eye movements and your eye movement plans. Where are you going to look next? Well, what we've determined is that this oculomotor circuit seems to be the most strongly recruited in these kinds of tasks that I'm describing, even though the animals are not trained to make eye movements. They're not reporting their decisions with eye movements. They're actually asked to keep their eye movements still at the center of a screen. Yet this network seems to be recruited in a way that we didn't anticipate to help solve these tasks. So one of the things that we've been thinking about is like, well, how do we rank all of these areas that we've recorded from and studied over the last 20 years in terms of their importance for this particular categorization task? And the ranking did not turn out the way we expected. Right? Areas in the early sensory system, they're just encoding the sensory features of the stimulus. Like, they're not important for the decision, except that they need to, do this basic background processing, but they're not really decision correlated. They're just encoding the features of the stimulus. Areas like MT, kind of less important in the decision process. These reaching related areas also seem less important. Even the dorsolateral prefrontal cortex. When we started out these experiments, we thought that was a very likely area to be contributing in a crucial way to these experiments because it's associated with higher cognition. It's not that cool in this task, right? It's kind of just in the middle. It's sort of, it seems to, and for a whole bunch of reasons that we've written several papers about. But then, areas, as we get more into this sort of spatial oculomotor attentional network, we find that these are the most important regions for driving these decisions, and I'll tell you why next. Yes? Mm-hmm. Yeah. It means if you not, yes, it means if we've done those experiments now, or we've inactivated each of these areas, these have the largest causal contribution to the animal's behavior. They also have the strongest encoding of the decision variables that, that correspond with the decisions the animals need to make. So and they also show the decision representations the earliest, following the onset of the stimulus. So everything is pointing toward these regions so far for all of those regions, or all of those reasons. Okay, so what I'll tell you today, these core spatial and oculomotor regions show the strongest involvement in these abstract decisions, and this is independent of their well-known spatial and eye movement functions. Like these, I'm not saying that these areas aren't important for eye movements, they're crucially important for eye movements, but they also have this additional function that I think has been not appreciated in representing cognitive variables that drive decisions. Okay, let's just zoom on here. So I'm going to show you what some of this data looks like, right? So I'm going to show you a few examples of individual neurons. These are single neurons that we've recorded from. 2 examples. Both of them are from area MT. This is that motion processing area that encodes the direction of motion of the stimulus when it's presented. Now, just to orient you, this is the firing rate and the number of spikes per second for each of these neurons. The motion stimulus is on here. This is the delay period when the animal's keeping in memory what he's seen. And what you'll see is both these neurons are quite active during the period of stimulus viewing. The 12 traces correspond to the direction of motion that the animal saw. So you can actually make sense of what these neurons are doing by just looking at their tuning curves. This neuron is most activated by a direction of 240 degrees, and activity just falls off as you move away. This neuron likes directions around 0 degrees. Again, it's tuned. Activity just falls off as you show directions further and further from the preferred direction. This is classic directional tuning in MT, just like what's been seen there for decades. So, but remember, the animals are performing this task, right? They are categorizing. They're saying this is in group 1, group 2. These neurons are not directly informative about that decision. They just encode what direction is the stimulus moving in. Notice when the stimulus turns off, activity just falls right back down to baseline levels. They're just encoding with it what the animal sees, what direction the thing is moving in. So this is why we would say a neuron like this is not particularly important for representing any kind of decision variable that corresponds to the animal's choices. Instead, it's just encoding what this we'd say it's a veridical representation. It encodes what the stimulus actually looks like. Okay, but then if we look downstream in the parietal cortex, this is just, this is an area that's directly interconnected with MT. We see a very different pattern. This is a neuron that shows a strong response to many of the directions, but notice it responds strongly to all the directions in one category, weakly to all the directions in the other category. And it's a binary-like response, right? If you needed to decode which stimulus in this category was shown, you're not going to do very well. This neuron is really showing a strong explicit representation of category. Now, this is a neuron. Well, sorry, I don't have that one here. We also see signals like this actually throughout this whole network of regions that I highlighted earlier. And our task has been to ask, well, which of the areas in this network seems most important for the task? One thing to point out, notice that neurons like this one don't just encode categorical decisions when the stimulus is on the screen, but they keep encoding that information during the memory delay, when the animal's sitting there in the dark, just keeping information in memory about what he saw in the past. Okay, so question then is what circuits and what computations transform the encoding of basic visual features, like in MT, into these learning-dependent representations of what the stimulus actually means to the animal, like in LIP. And so we've tackled this in a few different ways. Since this is NITMB, I wanted to tell you about sort of our computational modeling approach to doing this before sort of coming back to our latest results in the oculomotor system. Yeah.
Category that you're considering, like motion and stuff, or like most of the internet category?
Yeah, it's a crucial question, right? So we've done a number of experiments with other kinds of stimuli now too. So we chose motion because we have a good handle on how motion's represented, but actually what we're learning is that what we're studying here is general across probably any kind of visual stimulus we've shown. We haven't actually, just last week though, I was in Houston, and Jeff Yao, who has a monkey lab there, he's trained monkeys on a somatosensory touch categorization task where the animals had to group touch patterns into two categories, and he's finding the same kinds of signals just adjacent to some of the areas that we're looking at. So I think this is a general abstract decision signal, not one that depends on a particular visual feature. We have examples for shapes as well. So, but I think what's probably very important is the training, the explicit, training, the explicit grouping of stimuli into learned categories. Now, if it's just, if you mentioned ImageNet categories, I think it would depend a lot on what task the animal's performing. I mean, during passive viewing, if you were just showing images to the animal, even we know that we've done these experiments. These areas are engaged when the animal's actively performing a task, not when it's passively viewing images. So if it was, so it's quite different than IT, for example, which is kind of a mixture of feature representation and some semantic category information. Where? Yes, but there's relatively little, there's less of a feature representation of features that are irrelevant for category. It's like there's a representation of the, categorical membership at the expense of representing other features. Now, this is a very simple stimulus, right? One dimension, it only varies across one dimension. So we haven't been probing it with complex objects and things. So there's more we could do there. Yeah, do you have a question? Yeah.
What happens when these decisions are made because of the relationships? Like relationally, I'm wondering how the process. It seems to me like you're able to like locate things very specifically, but like aren't decisions made because there's sort of this is in relation to this, and so like that decision.
So you mean bringing together multiple lines of evidence or multiple stimuli or something like that? Yeah, that's an interesting question. And actually, we talk in the lab about how we might design tasks to study this. And actually some labs have studied sequential decisions where you might show a stream of different images and that the ultimate decision you need to make is based on weighting the evidence from each of these different stimuli. So these are the kinds of things that one could ask. In these experiments, I haven't really addressed that. We're really sticking with this simpler decision, category boundary-based decision in order to keep things simple, which we then hope that we can extend out to more complex decisions later.
Do they make a decision based on where that sits relationally to all the things?
Well, so one of the things you're bringing up is, what is the animal's representation of these categories and what is the strategy the animal's using? Does the animal have a conception of the prototype of each category, and what he's doing is comparing the direction he's seeing and how does it relate to the kind of prototype? Or is he doing, there are other ways of solving this too. Does the animal have a conception of where the boundary is and comparing to this direction and where does it sit relative to the boundary? We can get a bit of evidence actually for this. We can look at the animal's decision time. How long does it take him to make his decision based on different directions? You can get a little bit of, you can get some hints about the strategy the animal's using, but it's something you could also probe in human subjects too, where you can actually query them and you can actually test things more rapidly. But yeah, good questions though. Interesting. One of the things that we've been doing increasingly in the lab, because as you're probably getting a sense, training a monkey to perform a task like this or any kind of sophisticated behavioral task is a really major undertaking, right? It represents, some 100, 200 training sessions by, a graduate student or postdoc, to work with these animals, every day for, the better part of a year. You don't do that unless you have a really clear hypothesis built on a strong foundation, and it's not a great platform for doing exploratory science, right? And to test out just any hypothesis we might be interested in. But this is actually where working with artificial neural networks that can be now readily trained to perform tasks that are similar to the kinds of tasks we're studying in the lab, this is where these networks have become a really helpful approach for us to sort of think through our problems more deeply, to generate hypotheses and to test out the kinds of analyses that we're interested in performing on our real neural data. So we we often use recurrent neural networks in our studies. These are, rather than, many layer feed forward neural network architectures, recurrent neural networks are advantageous for the kinds of tasks we're interested in, because this recurrent layer can generate interesting dynamics, which can solve tasks which involve time, right? Which involve gathering evidence over time, which involve making comparisons between events that happen in sequence. So the way that we use neural networks in our research is really maybe different than in many situations, in that we're not really very interested in using this to fit our data. We're actually not fitting any data, neural data, with this model. Instead, what we're doing is we're training the models to perform the same kinds of behavioral tasks that we're studying in the monkeys. In a sense, we're using these models as a sort of experimental organism, right, that we are training to perform tasks, and then asking, well, how does the network solve the task? And we tend to choose networks that are based on very simple kinds of units, right? Rate-based units, which we define as being either excitatory or inhibitory, because we know in the real neocortex, in the mammalian brain, 80% of neurons approximately are excitatory, and that they only release excitatory neurotransmitters onto their targets. 20% of neurons are inhibitory. So we include that in our models. The way we set up many of our models is we will start with project input units, which encode, which are kind of mimicking the tuning that we know exists in the early visual system. So here what we have would be a pool of 36 motion direction tuned units. Each of those would be like those MT neurons that have a preferred direction, and activity falls off as you move away from the preferred direction. So that by activating some subset of that input pool, that would simulate a particular direction being shown to the network. So that input pool projects into the recurrent layer, and then the output units correspond to the behavioral responses that we would ask our monkeys to engage in. That would be reporting match or reporting non-match, right? Because that's what the network and the monkey need to do to solve the task. And then how are the networks trained? With backpropagation and gradient descent. Yeah.
Curious about the time constant, because that seems long to what time constants for individual neurons that I'm familiar with.
Yeah. The 100 millisecond time constant in the parietal, front, and prefrontal cortex. So actually, neurons, if you look across the hierarchy from early sensory areas up to areas in areas involved in more cognitive processes like prefrontal cortex, you'll see a progressive increase in time constant. So we're thinking about this input pool as basically being the output of the visual system. We're thinking about this recurrent pool as being the regions that we think are most involved in the decision process, which would be like parietal frontal areas. So this 100 millisecond time constant is not too bad of an approximation, actually, for those regions. So these kinds of networks, this is just what I already described. So we are training networks on this same task. So literally, we will have a input stimulus turn on for this amount of time, in terms of simulated time steps. We'll have a delay period where the network needs to remember the stimulus that was shown as the sample stimulus, and then the network is shown a second stimulus, and the network has to elevate its match unit if it's a categorical match, or elevate its non-match unit if it's a categorical non-match. And networks like these can learn to perform a task like this quite easily. Actually, this was the thesis work of Chai-Seng Mongkon, who has her own lab in Thailand now. And she was doing this work probably in 2014 or 2013 initially, kind of right when recurrent neural networks were really starting to be used more in neuroscience. She was very interested in asking whether, what she could learn by training networks like this on our tasks. At first, I thought maybe it would just be an exercise, and where would this go? But in her thesis committee meeting, I became very excited when she showed me this figure. So what I'm showing you at the top are 4 example neurons from our actual recordings from the monkey brain, right? These are two neurons from the parietal cortex. They show this categorical signal. You can see like the red traces are above the blue traces, or the blue is above the red. And these are two neurons from the prefrontal cortex. Now, what I'm showing you in the bottom, these are plots of the activity of 4 units from a trained network that's performing the categorization task. And what was so striking is that units from the trained networks would show category selectivity, but they would show it with similar dynamics, as we see in the real brain. And this was exciting because it suggests that, well, it was exciting just because it perked my curiosity. Right? It's like, well, here's a system that's performing this task that I've been studying for 15 years. It's doing so with activity that, at the single unit level is encoding the decision variable that is required to solve the task. Can we go in and understand how it's working better? This is when, the idea of using this as sort of a test bed for ideas really came about for me. In the hidden layer 100.
You chose 4. What did the others look like? Like, are these 4 representative ones? Are they sort of outliers?
These, they're not outliers in that the dominant signal that's being represented by that network, like especially during the delay period, the units are either encoding this category variable, or there are also units that seem to be participating in just keeping the activity level of the network in a high enough state that activity doesn't die out, right? Because activity needs to propagate across this 1-second memory delay. And that's where recurrent neural networks, are well known to generate attractor states that can do so. So the kinds of selectivity we see tend to be this category selectivity or just excitatory, activity that is keeping activity alive during that delay period. Yeah.
That biological networks were way more energy efficient than artificial ones because, but I mean, I know you're doing rates spiky, but I still had assumed like rates could get very small to represent rare spikes and low energy, but is that?
Yeah, it's a good, no, it's a good intuition. So this is, we actually, in other studies, not this study, we have actually explicitly put in sort of metabolic constraints where we've trained networks in a way that they are rewarded for solving the task with as little activity as possible, and that's fine. I guess in terms of energy efficiency, I mean, we're using a small network here. It's 100 units. We actually have a network. This is actually overpowered. If we went down to 50 units, it's still fine. I think getting below 30 and it's getting to be too little. So it's a pretty small system that's still able to perform this. Of course, we're doing a lot of the work for it, and that we're providing the processed sensory stimulus, right? Yeah.
Maybe get into this, but you have the anatomy for how the regions are connected. If you look at the connectivity in the model, is the order, the sequential order, and the similar units in the model, do they line up with the connectivity?
Yeah, that's a really interesting question. And, I think one thing you're getting at is whether, well, if you have a network like this, what is the structure? What is a hierarchy that develops? Does something develop in this RNN, in this soup, that actually starts to resemble the hierarchy that's in the actual brain? That's actually one of the things that we've been working on. I think it was actually the next slide I was going to show, is that so the answer, if you just have an unstructured network and just train it, it will solve the task in whatever efficient way it finds. And it will not, it doesn't mimic the biological hierarchy in any way that seems obvious in a simple network like this. But we've actually, in another study where we had a particular hypothesis about what the frontal cortex's role is in these tasks compared to the parietal cortex. And so we wanted to set up a multi-module RNN that would potentially test whether our experimentally generated hypotheses would actually play out in a system that was forced to be modular. And so the way that we basically split up our one pool of recurrent units into 2 modules, but in order to generate this modularity, we added in some constraints. the constraints we added in are that, okay, we have our input units, which encode the direction of motion. We still have our output units that record the decision. But we have constraints such that the input units can only project to this first module. They can't project to the second module. And furthermore, we also found that the network, just like our monkeys, the network is happy to cheat. So we found that, well, what this second module would tend to do is listen to those neurons that were getting the direct projection from the sensory units. And so it would be like just a reflection of the sensory input. So we added an additional constraint, was that the units that were directly receiving input from the input units could not project to the second module to like really enforce this modularity some more. And also the match and non-match units were only allowed to receive inputs from a subset of units in this higher order area. And so this is one way that we think about how to mimic some of the modularity that we know exists in the monkey brain and mimic it in these simple recurrent neural networks. You can imagine how this might scale up. I mean, of course, we could and actually one thing that we're doing, Pulkit Sinj, a grad student in my lab, is now implementing a sort of visual processing front end. So actually adding in, getting rid of this, pre-processed sensory representing stage and adding in one that more directly, that actually, functions as a motion processing system that can work on pixel-based input. So anyway, we're following this approach here, which has been really helpful in generating ideas and giving us the ability to test some hypotheses and ideas and analyses in networks where we know all of the connections, right? We know the anatomy, we know how these units are connected with one another. We can ask all kinds of things that we can't do in the monkey brain, where we can generate sparse samples of activity, but without knowing the underlying ground truth. So this is one of the ways that these kinds of models are used in our neuroscience experiments. So maybe just in the last few minutes, let me tell you about sort of where we are headed now. Because 5 years ago, we were very focused on the posterior parietal cortex, this area LIP, as being the best candidate for actually leading the decision process. We had done experiments where, we inactivate that area temporarily with a pharmacological agent and find that the animal's performance is degraded on the categorization task. But we started following the anatomy, right? We know that anatomical connections told us that LIP was very strongly connected with this network of areas that's normally thought, or it's known to be involved in directing attention and eye movements. So that's the frontal eye field and the superior colliculus. And we actually had projects in the lab at the time that were kind of focused on eye movements. We were interested in eye movement-based decisions, but we were starting to notice that in these tasks, there were much stronger cognitive signals than we expected. So we decided to pursue this more seriously. Okay, so these are the folks that really have done the work I'll tell you about today. Barbara Pesakovich was a PhD student in the lab. She's now a postdoc at Harvard. Oliver Zhu, he defended his PhD earlier this year. He's now back at the Pritzker School of Medicine. He's an MD PhD student. And Vinay is a postdoc in the lab who's still carrying on sort of follow-up experiments to this. So we became interested in this node in the primate eye movement system called the superior colliculus. So the superior colliculus is evolutionarily a very old structure. Actually, every vertebrate animal has a superior colliculus, right? The smallest fish, frogs, reptiles, birds, and of course mammals, monkeys, humans, we all have a superior colliculus. It's the area that's colored in blue here. But what you'll notice is that in these smaller brained vertebrates, superior colliculus is much larger compared to the rest of the brain. In fact, in these species like fish and reptiles, it's referred to as the optic tectum. It's actually regarded and appreciated to do the bulk of the visual processing of information coming in through the eyes and connecting visual representations with actions. These animals don't have a cortex, right? They have a simpler brain architecture, and the superior colliculus is known to be very important in all kinds of sensory motor behaviors. In vertebrates, and especially in primates, the superior colliculus is generally regarded as being involved in generating eye movements and head movements and orienting. Yeah.
They were all the same
Yeah,
state.
we have to look. I'm sure they're not identical. The brain of a macaque monkey brain is about that big. Mouse brain, what's a mouse brain? About that big? Yeah. So, even I could tell you that the macaque monkey brain is about the size of a pea. I'm sure that the colliculus in the mouse is probably a little smaller than that. Yeah. But proportionally, it's much smaller in mammals. But evolution does not throw away, previously existing circuits, right? It tends to build on top of them with new circuitry. And even though it's traditionally viewed as mediating kind of reflexive orienting movements of the eyes and head, recent work has started to appreciate that the superior colliculus is involved in some more complex behaviors, even some cognitive behaviors that involve eye movements and head movements. And so it was the path by which we just started to work in superior colliculus was a bit circuitous. I had a postdoc in the lab that had a lot of expertise working in the superior colliculus during his PhD, and it kind of motivated us to get some pilot data for a project he was thinking about. The monkey that we were working with was trained on this motion categorization task, like I told you before. It's an even more complicated and more challenging version where we actually have categories that contain directions that are in opposite directions, right? So this is really hard. You have opposite directions of motion belonging to the same category, and neighboring directions of motion that are often from different categories. So it took even longer to train the animals, but it's a nice design because it allows us to really carefully tease apart representations of direction from representations of category. But anyway, that's what the monkeys were trained on, the same kind of delayed matching task. And what was striking is even on our first recording session, when we started recording from the superior colliculus, we saw neurons like these that showed this pattern of category selectivity like we'd seen in our previous experiment. So a neuron like this one responded more strongly to directions that were in category 2. This neuron responds more strongly to directions in category 1. And they were big effects. And notice that this is a neuron that's showing that differentiation between categories when the stimulus is on the screen, and even encodes that information during the delay period. The first thing you'd think about is, well, wait, this is an eye movement generating area. Are the animals planning different eye movements for the different categories? Does that explain what's going on? And it does not. Remember, the animals are reporting their decisions with a hand movement, releasing the lever out here if the categories match. Instead, these neurons are showing these very strong category signals earlier on. We were recording simultaneously from the parietal cortex and superior colliculus in these experiments. What I'm showing you here is the strength of category selectivity that we see in one of our monkeys in the parietal cortex. This is the strength of category selectivity we see in superior colliculus. It's stronger and it's even shorter in latency in both of these monkeys, which was a big surprise. We really didn't expect to see. I would have expected that if category signals emerged in the colliculus, they would have maybe lagged that in the parietal cortex. Because remember, we had spent 15 years being very excited about LIP, thinking that was our best candidate for generating these signals. So this was a twist, and has made us reconsider what's going on. And then, okay, so this is a region that's showing strong encoding of these categories. Does it actually play a causal role? So to test this, we target the superior colliculus for a pharmacological injection of mucimol, which is a GABA agonist. It inhibits neural activity, temporarily shuts down activity in the colliculus for a few hours. And what we find is when we do that, the animals are dramatically impaired on this categorization task. This is really this was a very surprising finding to see such strong impairments on categorization. And it wasn't because the animals were just blind, because they could still perform other kinds of discriminations that were simpler. They could still release the lever. They were just specifically impaired on performing this categorical decision. Previously, we had found that LIP also contributed to these kinds of categorical decisions. This was the size of the behavioral effect that you see when you inactivate LIP. This is what happens when you inactivate superior colliculus. By the way, we used 95% smaller dose of mucimol in superior colliculus compared to LIP. So it's like a small perturbation of superior colliculus activity has a really big impact on the animal's behavior. So maybe, yeah, Lucas, yeah. Unilateral. And so so we see these effects when the visual stimulus is presented in the location that is contralateral. So where the stimulus would be in the receptive fields of the neurons we inactivate. Yeah. Yep. So the last thing I want to tell you about is that we've actually identified a sort of behavioral signature of the superior colliculus getting involved in these kinds of decisions, which is that when we looked very carefully, we looked at the period in this delay period when the animals are remembering what category they saw at the beginning of the trial. And what we found is that the animals actually made subtle eye movements during that delay period that actually betrayed what they were keeping in mind, right? Kind of like a poker tell, in a poker player or something. I can show you an example of this. What I'm going to show you is the animal's eye position throughout the delay period on trials where category 1 was shown and trials where category 2 was shown. So here's a movie, there we go. So here we're kind of at the beginning of the delay period, and we're looking at all the trials. These are the trials where category 1 was shown. These are the trials where category 0 was shown. So actually, these are very small. The animals are instructed to keep their eyes within this fixation window. There's a spot at the center of the screen, yet they make these very small, like half a degree or smaller eye movements that are very reliably encoding what category they're keeping in mind. They don't happen when the animals, they don't happen so much when the animal's looking at the stimulus. It's during this memory delay period when the animal needs to keep information in memory. And we see this, we actually brought together data from all the monkeys we've studied over the last 15 years, 11 different animals. And we find that all of the animals actually show some kind of little signature of these decisions in their eye movements, particularly during the delay period. But then some of you out there might be worried, well, wait, does this just explain all of your neural data? Because this is an eye movement area. And so we were, of course, worried about that. And the short answer is no, it really doesn't. So we can ask, well, how well can we read out the category, that the categorical decisions from the animal from neural activity in superior colliculus? And this is the time course of our ability to read out the animal's category judgments from neural activity in the sort of bright blue. We can also ask how well can we read out the animal's category decisions from their eye movements. Remember, they report their decisions with a hand movement out here, but we can ask, do the eye movements actually convey what category they saw earlier? They only do so later, like during this delay period, and you see that the activity about the neural activity related to category is very decoupled in time compared to the eye movements. And I know that we're basically out of time here. I think we're supposed to stop at 11 o'clock. Is that right? Yeah. So the last thing I'll just tell you is that what we've determined is that there is a unique subspace for cognitive information relative to saccadic information in the superior colliculus, where if you thought about a decoder sitting downstream from the superior colliculus population reading out left versus right eye movements, that would be projected on a subspace on a plane like this one, left versus right. Well, it turns out there's an orthogonal category subspace where activity based on category can, modulate up and down and minimally affect the representation of eye movements. And it's in the paper, but we've also shown that when the animals do wind up making these little cognitively modulated eye movements, it's because there's a transient alignment between the category subspace and the saccade subspace. So, and that happens in superior colliculus just prior to when these eye movements happen. So anyway, just to wrap up, we think we're excited because this work is kind of revealing to us that this system of the brain that is thought to be very primitive and is thought to be involved in these relatively simple behaviors like orienting your gaze, orienting your heads, is actually recruited to solve these kinds of very highly trained, cognitively demanding behaviors. And so just to summarize, abstract decisions are encoded widely across spatial and oculomotor areas with LIP and superior colliculus. And actually, I didn't tell you about the frontal eye field, but it's also very strongly engaged in this process. the superior colliculus and frontal eye field exhibit surprisingly strong and causally significant abstract categorical encoding, and category encoding in superior colliculus and FEF is independent of their roles in eye movements. And what we think is happening, which actually generates things like poker tells, is that there's leak from the cognitive signals in these spatial brain networks, which leak over to the subspaces which typically are driving movements. So thanks for having me today. I want to thank everyone that made this work possible. And you can see them all here and our funders. So thanks again. Oh, great. Oh, yeah. Have we looked into varying the EI ratio of the neurons? Yes, we have. We haven't really systematically varied it. But we do find that, adding in inhibitory units, we of course started with the ratio that we took from biology. That adding in inhibitory units actually helps stabilize these networks, and they're more trainable than when they're, purely excitatory, for example. But yeah, there's a lot of work that one could do following up on that. Yeah. Yeah.
How sensitive are the model-based results to the hyperparameter tuning and exactly like what's the ratio of excitatory inhibitor, like
Yeah.
units do you select for the result?
Yeah, using a powerful training approach like backprop, most networks, independent of their sort of the starting initialization, can learn to perform a simple task like this. Although we have worked with some more complex tasks and explicitly looked at the effects of initialization and actually asked, can we just find a better way to initialize networks? Can we, it's almost like what evolution has done for biological organisms, right? It creates this good initial state that can then, be useful for training. So we've played with that. We have a bioRxiv paper about it. It's something that's still kind of ongoing, but I think it's a really interesting question. Also adding in modularity, it should help, right? But it's also, there's a lot of ways to go about it, right? There's so many different architectures one could decide to explore. So I don't know, the exercise of playing with these different things have been really helpful for us just in our thought process, but I don't know if I have anything really deep to say about how to choose the right parameters, other than to say that we're probably most excited about choosing parameters that match what we would expect from biology.
It's not that you're experiencing like stark robustness to these choices.
Not in a way that's very obvious, other than, for simple tasks like the one I've described, like our main categorization task, it's not a very challenging task for these networks to learn. So I think you maybe need to challenge the networks. We've also, we've actually done a lot of work where we have like a whole battery of tasks, right? Where we train the networks on maybe 10 different tasks, including variants where there's, flexibility, where you might have in one, context, the animal or the network might need to perform, like solve a task based on one rule, in another context, they have to solve it based on another rule. And that becomes more interesting and more demanding for the networks. Great. Well, thanks, everyone.